Hi everyone, and welcome to our fourth Biology Daily Booster. Notice the big purple box is back because we are still continuing with B1.2, which means this is guaranteed to be questions on your first biology paper this year. What we're going to be doing in our booster today is having a look at enzymes. So, first of all, we need to know that enzymes are proteins. Now, that means they're made from amino acids because a protein is a polymer made of the monomers of amino acids, and they're going to fold up in a very specific shape. Now, the shape is important because there's a region on the enzyme that is called the active site. Now, the shape of the active site is complementary to the substrate, the thing it's going to join with. If the active site doesn't match the shape of the substrate, just doesn't work. So the active site is important. Know the name, know what it looks like in a diagram. It's basically a little Pac-Man diagram. I will try and draw it, but I only have my mouse today, so I apologize, this will be terrible. So there's our little enzyme, and then the active site has a very specific shape there. So your substrate would have to be complementary to this little shape here. The other phrase we use as far as enzymes go is the words biological catalyst. Now, biological because they're in living things, and catalyst speeds up reactions. So the phrase biological catalyst means it speeds up reactions in living things. Because the active site matches to the substrate, then what we can say is that enzymes are specific. They will only bind to a specific substrate because the active site and the substrate have to be complementary. We can explain this with something we refer to as the lock and key hypothesis. And by that, what we mean is if you think about a lock and a key, the key is the complementary shape to the individual lock. So one key will open one lock, but it won't open every lock in the world. Same idea with the enzymes. When the substrate joins to the active site of the enzyme, we make this thing called the enzyme substrate complex. Now, at that point, that's where it's catalyzing the reaction. It could be joining things together, it could be breaking them down, but whatever is happening happens, and then the products are released from the enzyme. That means our enzyme is free to then repeat that same process with new substrates. Because it's a catalyst, it is unchanged at the end of the reaction. We need to know about 4 factors that are going to affect the rate at which enzyme catalyzed reactions occur. They are temperature, pH, and then the concentration of the enzyme and the concentration of the substrate. This is going to be an important section because one of the assessed practicals for the higher tier folks is about enzymes. So we'll talk about that as we look at the actual effects. The first one then is temperature. So on the left you can see the graph of temperature, and the right gives you a bit of an explanation. The key principle is, in this first part of the graph here, as we increase the temperature, you can see the rate of reaction increases. The reason for that is that as we increase the temperature, the particles gain kinetic energy, they move faster, so they have more frequent collisions. We then hit this point at the very top of the graph, which is our optimum. So the optimum is where the reaction is happening at its fastest possible rate. Depending on the enzyme, the optimum temperature may be different, but when we're thinking about enzymes in humans, optimum temperature is around 37, which is human body temperature. When we go beyond the optimum, so if we keep heating up beyond that point, then the actual temperature gets too high. And at that point, we're going to denature the enzyme. So this word denature is important. When we denature the enzyme, that means we've changed the shape of the active site, the substrate no longer fits, and therefore it can no longer catalyze that reaction. So, what could they ask us in terms of a practical? They could give us a setup of a practical here to do with enzymes and ask you things like how we could change the temperature or how we could improve changing the temperature. They may give you an example where it's a water bath which is set up with a Bunsen burner. Really terrible. The improvement would be to use an electric water bath, because we can set them to a specific temperature and it keeps it at that exact temperature. Plus, no flames, therefore you're not going to set yourself on fire. So there's no kind of fire risk by using an electric water bath. Number 2 of our factors is pH. Now pH is quite straightforward. All enzymes have an optimum pH, the pH at which they will work at their fastest rate. That optimum will be different in different enzymes, and it depends on where they're designed to work. So enzymes that are designed to work in your stomach will have quite an acidic optimum pH because your stomach is acidic, but one in your mouth would not be acidic as its optimum. If we go too acidic or too alkaline, then we denature the enzyme. Then in terms of our substrate concentration, what we actually find is initially increasing the concentration increases the rate of reaction, but we hit a point where increasing substrate concentration does nothing to increase the rate. And that's because at this little point here, the little X, that is our point of saturation, which just tells us that all of the active sites are occupied, therefore it can't go any faster. In terms of increasing the concentration of the enzyme, what we see is just an increasing rate of reaction, because the greater the concentration of the enzyme, the greater the rate of reaction will be. Now, we did briefly mention the PAG on our enzymes, but there's a couple of other things we're going to cover here. So, they could ask us how to improve a method. Typical ways are listed on the left there for you. First one is suggest that we use smaller intervals between readings. What I mean by that is if we're going up in 10 degrees, for example, so 10, 20, 30, and we can see the optimum somewhere between 30 and 40, we don't know exactly where between 30 and 40. If we were to do smaller intervals, so maybe every 5 degrees, every 2 degrees, we get a more accurate picture. Option number 2, do more repeats and take the mean. Please get into the habit of saying do more repeats and take the mean. Take that as one sentence that can't be broken up into 2 sections, because you will find that quite often the mark to get there will have both parts required. Third one is to get rid of human judgment. A lot of the enzyme reactions rely on colour changes, and we are quite rubbish at judging colour changes as human beings. So we use this bit of equipment called a colorimeter instead of the human judgment, so it's objective, not subjective. And we've already mentioned the reasons why we use the electric water bath: control the temperature or set it to a specific temperature, and limited fire risk. So do make sure that you are aware of those key parts on the method. Other than that, you could be asked to draw a graph, you could be asked to calculate means, but I can't really think of many different ways they could phrase the PAG on the enzymes. I may be wrong, I hope I'm not. As always, final thing to do for today is to head on over and have a go at the quiz to check your understanding of today's work. So the link is again in our description, so you can hopefully find it nice and easily. Don't forget to come back tomorrow where we'll continue with our biology daily boosters.